DNA is the fundamental building block of all complex life forms. From birds to worms, it contains instructions for every cell in an animal's body. However, most cells don't need to follow every single one of these instructions, so certain genes will either need to be turned on or off. The process of turning off unwanted genes is called gene regulation and is handled by complex, three-dimensional proteins. Deep Genomics, a company focused on the intersection between artificial intelligence and gene editing, has found a way to use convolutional neural networks to predict what sequences of DNA these proteins will attach to. To train this model, an exorbitant amount of data was needed. Millions of sequences were collected from different sources. These sequences are made up of 14 to 100 different letters, each representing a corresponding molecule. Because the information in DNA is made up of just 4 molecules, adenine, cytosine, thymine, and guanine, a sequence of DNA can be represented by a list of A's, C's, T's, and G's. After giving the AI model this collection of sequences, the convolutional neural network will look at collections of letters and try to predict what proteins will bind to them. By repeating this process millions of times, we can ensure that the AI model will be able to create an accurate picture of the real world. Once it's done training, we can be confident that the AI model will be able to accurately predict what proteins will bind to a given set of DNA. By being able to accurately predict what proteins will bind to a specific sequence of DNA, we will be able to develop many new genetic medicines at a much lower cost. Aging, as an example, is strongly correlated to a loss of gene expression, and it therefore might be possible to stall or even halt this process through the use of these proteins.